Welcome to topic 7-2. This is the last lecture for the course, and we're going to be covering meiosis, which is a condition in animals that's brought about by flies, and primarily by the three fly families shown here, the Calliphoridae, Sarcophagidae, and the Estridae. Now, given that this topic is focused on a condition, we will not use our typical boilerplate for the lecture. So the learning goals you'll see are going to be a little bit different than what we've been using for the past several lectures. So instead we're going to focus on what this condition is, as well as the group of flies that cause it, what is necessary for meiosis to occur, and then lastly, what is the veterinary importance of meiosis. So let's start with learning goal number 1. So what is meiosis? And this word literally means fly condition. So maia, which is Latin for fly, and osis, which is condition. And meiosis can be defined as the invasion of a living vertebrate by dipteran larvae. And these larvae feed on living and or dying tissue of the vertebrate as a source of protein. So we've talked about dipterans that feed on blood as a source of protein. In this condition, the dipterans are feeding on a living vertebrate, so feeding on the flesh of that vertebrate, whether the flesh is alive or is dead. Now, you may have heard of this condition in other terms. So it's sometimes referred to as fly strike or fly blow as a common name. And you can categorize meiosis by the degree of the relationship with the host, as well as the site of invasion or infestation on the host. And in general, there's three categories of meiosis, and they're referred to as accidental, facultative, and obligatory. Accidental meiosis occurs when a non-parasitic larvae are ingested, and this leads to harm or discomfort of the host. So this is a non-specific condition, and it's driven primarily by random events. So the flies are not seeking a vertebrate host. They're either accidentally ingested, so the fly larvae may be living in a food source for a vertebrate host, and then the vertebrate host ingests them when it wants to consume its food. But this is not a normal part of the fly's life cycle. And we're not going to be spending a lot of time discussing this accidental form of meiosis. Instead, we'll be focusing on these latter two. So facultative is characterized by free-living larvae that feed typically on carrion, so rotting corpses of dead animals. But they can also invade living hosts and become parasitic if given the opportunity. And then lastly, obligatory meiosis is the most specialized relationship of these three, in which the parasitic larvae require a living host to complete their life cycle. Now, in regards to invasion site, a meiosis can be categorized by which organ or tissue is primarily affected by the larvae. So here are some examples below. So this is an image we've seen previously, but this would be characterized as an ocular meiosis in which the eye socket is infected, a gastrointestinal meiosis in which we have larvae that are living in the gastrointestinal tract, or in this case the stomach. And then another example would be a genital meiosis, in which the fly larvae are living in the genitals of the animal. Now, some of you may find this phenomenon of feeding on live tissue to be quite striking, and wonder why in the world this type of life history would have evolved. For instance, why not just focus on developing and feeding off of dead animals as opposed to a live host? So this slide, I want you to think about some of the potential reasons that this strategy may have evolved in dipterans. And to help you think about these reasons, I've put up these four terms here: competition, availability, predation, and environment. And what I'd like to do is discuss each of these factors and see if we can come up with some possible explanations as to why feeding on a live, living vertebrate host would be beneficial or more advantageous to feeding on a dead vertebrate. So let's first tackle this issue of competition. And so by competition, I'm thinking of resource competition. So a dead animal offers no defenses, and it's highly susceptible to any animal or microbe that wishes to consume it. In contrast, a living animal is going to be a much more difficult resource to exploit. And so if you think of this, if you can potentially overcome the host's defenses while it is alive, there is going to be much less competition for that resource. So a dead animal is fair game to anything that can exploit that dead animal. The dead animal is not going to run away. It's not going to fight back. And so it becomes a free-for-all for microbes or other scavenging insects to feed on that animal. But a live animal there's going to be much less competition for that living animal's tissue if you can exploit it. And so that is one potential advantage is a resource competition. You can exploit a niche that has not been taken advantage of before by many other groups of organisms. Well, now let's talk about availability. And again, thinking in terms of resource availability. So finding a dead animal, I should say, is a rather unpredictable event. Dead animals can die just about anywhere or anytime or anywhere. It's not very predictable. In contrast, live animals have predictable behaviors, aggregation sites. So, for example, we've seen watering holes, we've seen food sources, nests, dens. These are areas where living animals, and particularly vertebrate animals, are going to congregate. And they're going to be fairly abundant in these locations. So, in contrast to dead animals, which have to be found and are rather randomly dispersed throughout the environment, living animals are a more reliable resource if, again, you can take advantage of them. If you can get around their defense systems and their defense behaviors, you have potentially access to a resource that is more readily available than a dead animal. Thirdly, think about predation. So invading a living host offers some sort of protection to the parasite. So invasion of a living host offers some sort of protection from predation to that animal, to that parasite that is living on the living vertebrate. So living in carrion, so a decomposing animal, you are exposed to a variety of microbes, other insect larvae, scavenging animals that may be just as likely to eat you if you are a small insect larvae as you are to eat that dead animal. However, if you are living inside the tissue of a living vertebrate host, you are protected twofold. So one, by the animal's skin, which is a barrier, and secondly, by the animal likely avoiding another animal that would be attempting to prey upon you. So by invading this living vertebrate host, these dipteran larvae are offered a secondary form of protection from other predators, where if they were living in a dead vertebrate that's decomposing, they're going to have a greater chance of being preyed upon secondarily by other scavenging animals. And lastly, let's talk about the environment. So living in a live host provides the dipteran larvae with a constant temperature, moisture or humidity, constant pH, so acid-base balance, and this living environment is sheltered from the elements. So if this is an animal that lives in burrows or lives in barns where it can be protected from rain and extremes in temperature, that parasite is going to be buffered from those extremes in temperatures. And so by providing a more constant environment, this may enhance the development of the dipteran larva. And in regards to temperature, think about most vertebrates, at least most domesticated animals, have constant body temperatures, and in cold months those body temperatures are going to be warmer than the external environment. So by exploiting a living vertebrate host, this may enhance the development times of those dipteran larvae by living in an endothermic animal. And you have to contrast this with the condition in a rotting corpse, which is going to be susceptible to any fluctuations of temperature, humidity, rainfall, pH in the environment, and whatever chemical changes occur in the tissues of the corpse or carrion that the dipteran larvae are living in as it's being decomposed and metabolized and digestive enzymes are being secreted into it, etc. So a dead vertebrate offers a much more variable and changing environment than that of a living vertebrate host. So if we take all of these factors into consideration, you can see how there may be a substantial selective advantage if these dipteran larvae can overcome the host defenses. So that is the primary barrier to living on a vertebrate host that is alive, is being able to overcome those defenses of the host, either the behavioral or the physiological defenses, such as the immune system. So continuing on with that line of thinking, let's now take a look at some of the typical anatomical features of larvae that are capable of causing miosis. And note that such larvae that cause miosis are often referred to commonly as maggots. So these are dipteran larvae that are capable of invading living flesh or dead flesh. And as you can see from this image here, these dipteran larvae are worm-like in their appearance, but they do have distinct anterior and posterior regions, which correspond to the head end and the tail end of the animal. And as you may expect for an animal that needs to invade living flesh, their mouth parts of maggots are specialized for feeding on flesh, and in particular they have these fang-like mouth parts. So if you take a look at this image on the bottom left, you can see these just really scary looking fang-like structures coming out that are the animal's mouth parts. And these mouth parts allow the larvae to grab onto living tissue and tear into it. And this mouth of the animal often secretes digestive enzymes that facilitate the softening of the living tissue and allows the fangs to work more efficiently. So think of this as a two-step process where the fangs are grabbing onto the flesh, then the animal is secreting digestive juices onto the skin of the vertebrate host, and then the fangs are clawing out that digestive tissue, facilitating opening up a wound site, and that will eventually allow the larvae to invade the tissue. And on the right you can see these fangs as well. These are dark black structures on this individual. Here are the mouth parts. Another distinctive feature that is observable in these images are rings of spines that go around the circumference of the animal. And these rings of spines are found throughout the axial length of the animal. So you can see they occur near the mouth parts, and also as you extend down towards the posterior region, there are these lines or rings of spines. And these spines resemble the dermal scales on shark skin, if you're familiar with those structures. And that is, they're oriented so that the larvae can move forward, so they can advance forward, but they are restricted in their ability to move backwards if they are embedded in a structure. And this is an adaptation that allows the maggots to resist being pulled out of a wound, as we're going to see later. So here again on this image you can see the orientation of these spines. So note that they project posteriorly. So they're small spines, but they're projecting backwards. And lastly, I just wanted to point out another interesting structure on these animals, which is at the posterior or back end of the animals. And on this posterior end, the maggots have distinct spiracles. So these ring-like structures and chambered structures you can see here. And these are chambers, these spiracles act as gas exchange surfaces. And I wanted to pose the question as to why these animals breathe with their back ends. So, what is it about the lifestyle of these maggots that makes it advantageous for them to have their breathing structures on their behinds? And the answer is that, remember, these animals are going to be burrowing into the flesh of a living organism. So this anterior end is going to be buried in tissue, and so but the back ends are going to be exposed to the environment. So that's where the air is going. They're going to have a chance to breathe air through their back end because their front end is going to be buried deep into the tissue of a living host. So that's why it's advantageous for these animals to have their breathing structures at their back ends, because the head end is going to be burrowed in the vertebrate host. Now, before we finish this learning goal, I wanted to review the three main taxonomic families of dipteran that are primarily responsible for facultative and obligatory meiosis in vertebrates. And those three families are the Callipheridae, the Sarcophagidae, and the Estridae. The Callipheridae includes species of screwworms, blowflies, and bottleflies. And these flies can have either facultative or obligatory meiosis with vertebrate hosts. And I should point out this is what the adults look like, not the maggots that would be invading the tissues. The Sarcophagidae are known as flesh flies, and there are representative species from this group that form facultative or obligatory meiosis. And then lastly, the Estridae, which are the botflies and cattle grubs. These represent the most specialized group of meiosis flies, and they only include obligatory species. And that means that these flies are completely dependent on invading living hosts to complete their life cycle. So without a living vertebrate host, these types of flies are unable to complete their life cycle. And that brings us to the end of learning goal number 1. So now that we've described what this condition is and which flies cause it, what is required for a cycle of meiosis to occur? And you'll note that this learning goal is similar to one we had back in module number 1 when we talked about arthropod-borne diseases. So in that lecture we asked what is required for a cycle of vector-borne disease. And so we're going to tackle this learning goal along a similar conceptual framework because, in essence, meiosis is an arthropod or vector-borne disease. And so for any vector-borne disease, remember what are those three requirements. So we need a pathogen, we need a host, and we need a vector. And of course, in this condition, the host in meiosis is a vertebrate animal. But I want you to think about who is the pathogen and who is the vector in this condition. And so before we answer those questions, I want to review a typical life cycle for meiosis, and then we'll discuss the answers to these two questions. And this figure below shows you a typical life cycle for meiosis. And let's start this reviewing this life cycle by looking at the adult female fly, which can lay its eggs or first stage larvae, if it's a live bearing fly, either directly on the vertebrate host or, interestingly, on the body of a blood feeding arthropod. such as a mosquito shown here. And then this mosquito would transport them to the skin of a vertebrate host. So there's two options for getting the eggs or the larvae to the vertebrate host, either directly by the adult female or through attaching the eggs to a blood-feeding arthropod, which then brings them to a vertebrate host. Now the larval stages of these myisis flies will feed on the host for a total of 3 larval stages or instars, acquiring protein and increasing in body size as they develop. The damage that the larvae cause to the tissue and the following necrosis or death of that tissue can often attract additional myisis flies to the wound sites, and then this stimulates those flies to lay additional eggs, causing what's referred to as a secondary myisis. So this initial laying of eggs or larvae on a vertebrate host is referred to as a primary myisis, whereas any additional myices that develop from that first infection would be referred to as secondary myisis. And this can lead to the development of a positive feedback loop, where as you get more and more flies laying eggs and larvae on these necrotic tissue, you get more attraction, you attract more and more flies that will lay more eggs, and this can just escalate out of control for the vertebrate host. Now once the larvae mature and fully develop on the host, they will fall off of the host and then pupate, usually in the soil. And then once these adult flies emerge from the pupal cases, they will often focus primarily on reproduction. So they've acquired all the protein they need from when they were in the larval stage feeding on the animal tissue. So when they become adults, they can just focus on reproduction. So with that general overview of a myisis life cycle, let's discuss what is the pathogen, or who is the pathogen in this life cycle. And the answer is the pathogen would be the fly larvae or maggot, which is responsible for the disease condition. So the larvae is causing the disease state. So it would be considered the pathogen in this transmission cycle. And then who is the vector? So who is responsible for transmitting this pathogen to the vertebrate host? And there are two potential options for this. So one is the adult fly, which could transmit them directly to the vertebrate host. The other potential vector is a blood-feeding arthropod. So if the adult female lays eggs on a blood-feeding arthropod, then this arthropod becomes the vector for the myisis. And to continue this analogy, what type of transmission cycle would you define this myisis disease as? So remember, we've talked about cyclodevelopmental, cyclopropagative, propagative, mechanical modes of horizontal transmission. Which one does this seem to best fit with? And I'm not sure if there's a strict or correct answer to this. I would argue that it lies somewhere between a mechanical and maybe a cyclodevelopmental transmission. So there's certainly a mechanical transmission of the pathogen, which in this case could be the eggs and larvae by the blood-feeding arthropod to the host. Or if you consider that the eggs need to hatch either on the vertebrate host or within the insect vector, then you could consider it a cyclodevelopmental transmission. But it's probably least likely to be considered a cyclopropagative or a propagative one. And so that concludes learning goal number 2. Now we're going to move on to the last learning goal, and what is the veterinary importance of myisis. But before we move on there, I think I'm going to stop the recording because the last section will take quite a while to finish up. So I'll stop the recording now, and then I will continue with part 2.